Hello, this is Anuja Matthew. In today's lecture, I'd like to talk about potential risks associated with vaccine development for COVID-19. So in individuals who get a vaccine, can you actually get more sick? In the previous lecture, I talked about different kinds of vaccines, RNA-based vaccines versus DNA vaccines versus live attenuated vaccines and protein-based vaccines. Each of them have unique strengths and weaknesses, but in all of the cases, the hope is you generate strong immunity so that you will get protected from a re-exposure to SARS coronavirus 2. But in a scenario where you generate antibodies that are not neutralizing, for example, you can actually cause disease through enhanced viral replication. So the virus actually replicates to higher levels in the presence of these antibodies. And I'm going to talk about that in the next slide. Another scenario that's mediated via antibodies that's vaccine associated is you might actually get enhanced respiratory disease that's based on antibodies that don't perform as well as they should, and they form immune complexes. Now, looking at the other arm of the immune response, which is the T-cell arm of the immune response, if you generate an immune response that's suboptimal or biased, this Th2-based immune response has been associated with ineffective vaccines. You can also get enhanced disease after subsequent infection. So as we've discussed in many lectures, ideally you would generate strong neutralizing antibodies. And what does a neutralizing antibody do? In the case of SARS coronavirus 2, this antibody, through its FAB portion, will bind the receptor binding domain of the spike protein and prevent the spike protein from attaching to the main receptor, which is ACE2 in the cell. By blocking the receptor binding domain on the spike protein, it prevents the virus from entering the cell. So the hope for every vaccine is to have long-lasting neutralizing antibodies that are able to prevent infection. You generate antibodies of all kinds of specificities and all kinds of effector functions. So many antibodies that you potentially generate against SARS coronavirus 2 may not neutralize. Or if over time the quantity of neutralizing antibodies decrease, what happens is these antibodies bind different portions, say, of the spike protein, and through its Fc, or the constant region of the antibody, it actually binds receptors on the cell in addition to the virus directly binding the ACE2 receptor. This is an enhanced opportunity for the virus to bind another receptor on the cell. So the antibodies actually help virus get into the cell more quickly. So instead of preventing infection, it's actually enhancing infection. And that's a deleterious effect. So this has been shown for many virus infections, including the virus that I study, which is dengue virus. So dengue viruses have been shown to enter monocytes and macrophages, which are antigen-presenting cells. They can enhance infection, or they come into antigen-presenting cells and replicate to larger quantities. And so, in a sense, these antibodies are actually causing virus to replicate more, and that's a bad thing. We just talked about antibody-dependent enhancement, whereby non-neutralizing antibodies bind virus, and through activation of the Fc receptor, virus-antibody complexes are taken in, resulting in productive viral replication and release of more virus. So in essence, you're enhancing replication because of the presence of these non-neutralizing antibodies. Another way where there's possibly immunopathology is the activation of antibodies binding the virus. They activate a certain pathway known as the complement pathway. So the virus and antibodies form these large immune complexes, activate the complement pathway. They enter cells through complement receptors, and because of this activation process, they result in the release of these molecules known as cytokines or chemokines. These are chemoattractive substances that can be released and can be actually quite harmful if produced in very large amounts. So this is one way in which you can get enhanced disease pathology. Another possible function of antibodies is antibody-dependent cell-mediated cytotoxicity. And we don't know yet if any of these functions actually work for SARS coronavirus, yes or no. This is theoretical at this point. But antibodies can also eliminate target cells or infected cells via this process known as ADCC. And this might be actually a beneficial thing, and we don't know that yet. The final scenario I'd like to discuss today is T-cell mediated immune pathology. Here, if the vaccine induces cells or skews them to secrete a certain subset of cytokines, and in this case we're talking about Th2 biased cytokines, then it's possible that these Th2 cytokines can actually cause increased airway inflammation, more mucus production, lead to cell death and airway wall edema, leading to enhanced or worsened disease. And there's some tragic experience with children who received a formalin inactivated RSV vaccine, which is the respiratory syncytial virus vaccine. And these children who received the vaccine had a far higher incidence of severe disease compared to those children who did not receive this formalin inactivated vaccine, and as a consequence of natural infection following vaccination. The most significant incidence occurred in the youngest children. And when they did autopsy studies of these children, in the airways, they found a large number of cells that were associated with these Th2 cytokines. So this was a very tragic experience of a vaccine gone awry. And this delayed vaccine development for RSV for about 30 years because of this experience. I'd like to end today's lecture by saying there's no evidence to suggest that a SARS coronavirus vaccine might induce ADE or vaccine-associated enhanced respiratory disease. For ADE, typically, viruses replicate in macrophages or monocytes. And for SARS coronavirus 2, the typical cells that they replicate in are respiratory epithelial cells and not monocytes, macrophages. They're very different types of cells. So it's entirely possible that ADE does not occur for SARS coronavirus 2. This is just a consideration and to keep in mind. In terms of vaccine-associated enhanced respiratory disease, it's not known yet. People will work on it and find out if there's significant immune complex formation. There are ways to mitigate the response or reduce the possibility that you can get worse disease or enhanced disease. So if you develop a vaccine by using conformationally correct antigens and in the process elicit high quality neutralizing antibodies that last a long time, then you will not have either ADE or enhanced respiratory disease. That's vaccine associated mediated by antibodies. Also, if you have, say, a DNA vaccine that typically biases cells towards a Th1, which is the better subset of cells and cytokines that are secreted and is associated with an effective vaccine response, then perhaps you will decrease the possibility of having a Th2 biased immune response. So I just thought that it would be helpful for people to think about potential scenarios where things could go wrong. This is why vaccines take a long time to develop. This is why vaccines have to be tested rigorously. And the furious pace at which we are going gives researchers some concern that we might be overlooking some possibilities. So thanks again. If you have any comments or suggestions, please do let me know.